Hi students, today we are going to learn about nerve muscle physiology. In that, now we are going to learn about nerve. In this, we will discuss about these three headings in this video. That is, first one is the functional anatomy of the nerve and biological activities of the nerve and then physiological properties of the nerve. Okay, so these three we are going to discuss in this video. First, coming to the functional anatomy of the nerve. Okay, functional anatomy. The nervous system is actually composed of two main cell types. What are they? They are neurons or nerve cells and supporting glial cells. Okay, these neurons are the structural and functional unit of the nervous system for both central nervous system and peripheral nervous system. So what is the basic function of the neurons? They will perform four main tasks. What are they? First one is they will receive the signals. Second one is they will integrate these signals. Third one is they can generate the signals. And fourth one is they can transmit the signals to the target cells and end organs Okay, so the nervous system is composed of neurons and glial cells. The neurons are the structural and functional unit of the nervous system, and these four main functions are performed by the neurons. And this neuron is having four main functional parts to perform these functions. So what are they? First one is the receptive part. It is present on the dendrites. These will receive and conduct the electrical signals toward the cell body. Second one is the integrative part. It is present in the cell body or soma. It contains the nucleus and most of the cell organelles, and it acts as the trophic center of the entire neuron. What happens here? Here inputs are processed and integrated to determine whether an electrical impulse, that is action potential, should be generated or not. So it will take the decision. Third one is the conductive part or axon. It conducts the electrical impulses away from the cell body. The fourth one is the transmissive part, that is axon terminals. These axon terminals communicates with the other neurons or effectors. Effectors means the target structures which respond to nerve impulses. So these are the four main functional parts. First one is the receptive part, second one is the integrative part, third one is the conductive part, fourth one is the transmissive part. See here in this picture it is depicted parts of a neuron with its functions. As we have seen in the last slide, the dendrites, it will receive signals from the other cells. Cell body, which is containing nucleus. Okay, this nucleus controls the entire neuron. The cell body organizes and keeps the cell functional. And coming to the axon. Axon is having axon hillock, then axon, and it ends in the axon terminals. In this axon hillock, the impulse will be generated. That is, action potential will be generated. Now this is the axon. It transfers signal to the other cells and organs. This is axon terminal. It forms junction with the other cells. This is the myelin sheath. We are talking about the peripheral nerve here. It is formed by Schwann cells in the peripheral nervous system. Why this myelin sheath is important? It increases the speed of the signal transmission. Here the Schwann cells will produce the myelin sheath in the peripheral nervous system. whereas oligodendrocytes, they will produce the myelin sheath in the central nervous system. And this nerve is covered by cell membrane that protects the cell. This is about parts of a neuron with its functions. Now coming to the first most important one, the cell body of a neuron. It is also called as soma. Where it is located? It is located at the center of the dendritic tree, especially in the multipolar neurons. What is this multipolar neuron? We will come to it later. Okay. Mostly the cell body or the soma is located at the center of the dendritic tree. Okay. Whereas the cell body is a spherical or polygonal in shape. So the cell body can be spherical or polygonal in shape. It is located at the center of the dendritic tree. It contains nucleus, and nucleus contains the genetic material, that is DNA of the cell. And also the soma contains cytoplasmic organelles such as endoplasmic reticulum, both smooth endoplasmic reticulum and rough endoplasmic reticulum. And it also contains free ribosomes. Okay. So these free ribosomes along with the endoplasmic reticulum, they will form nissal bodies or nissal substance. Okay, it is a chromatophilic substance. So this is involved in the protein synthesis. Okay, it is important for the production of enzymes, receptors, ion channels and other structural components. Next, coming to the other cell organelles, that is Golgi apparatus and microtubules. These are also present in the soma, and these are involved in the packaging and transport of proteins. Now coming to mitochondria, it is present and it is involved in energy production. And lysosomes are also present. These will manage the waste material in the cell. So what are the cell organelles present? Endoplasmic reticulum with free ribosomes. If the ribosomes attach to endoplasmic reticulum, it is forming nissal body. That is nothing but parts of the rough endoplasmic reticulum. They are called nissal bodies here. It is important for protein synthesis. Now, Golgi apparatus and microtubules for packaging and transport of proteins. And for production of energy, we need mitochondria. And for waste management in the cell, lysosomes are present. Now coming to the axon hillock. Axon hillock, that is the starting part of the axon. It is the site of origin of the axon. It is cone shaped and it is not having any cytoplasmic organelles. But it contains high density of voltage gated sodium channels. Okay. So because of the presence of high density of voltage gated sodium channels, it can generate, that is it can initiate the action potentials. It can generate the action potentials. And it also supports the neuron polarity. How? By separating the receptive and integrative parts from the conductive and transmissive parts. That is, receptive and integrative parts are on the one side of the axon hillock, that is dendrites and soma. And conductive and transmissive parts, those are axon and axon terminals. They are separated. So the polarity is maintained. The dendrites and soma are negative. We will consider that as minus sign. And these two, axon and axon terminals, are considered as positive end, negative and positive end. These two are separated by axon hillock. So it provides the directionality in the flow of information from the dendrites to the cell body and from cell body to the axon and axon terminals. So the directionality is maintained, provided by the axon hillock. Now coming to dendrites. These dendrites are the tree-like processes which are extending from the cell body of the neuron, and these will also contain similar organelles such as in the cell body, and these are highly branched structures. So why they are highly branched? What is the advantage of this? Because of this, they will provide increased surface area. So surface area is increased because of highly branched structure. So it is important for receiving the information from other neurons. Okay, and particularly at the synapses. So they are specialized areas of contact. So on the dendrites, we have specialized areas of contact to receive the information. Those specialized areas are called synapses. Okay, and in some dendrites, we have dendritic spines. Their dendritic spines are numerous tiny protrusions. They are called dendritic spines. So why they are important? They serve as the sites for initial processing of synaptic signals via membrane-bounded neurotransmitter receptors. So dendritic spines are important. Why they are important? They will process the information initially. In that, it is having some neurotransmitter receptors. Okay. So in the membrane of the dendritic spine, we have neurotransmitter receptor. These will initially process the synaptic signals. Okay. Then they translate the chemical messages which are received into electrical events. So what is the importance of these synapses and the spines? They will translate the chemical messages which is received on the dendrites into electrical events. Then these electrical events travel down the dendrites. Now coming to the axon. Axon is also known as nerve fiber actually. The membrane of an axon is called axolemma and the cytoplasm is called axoplasm. So here you have to focus on tract and fascicles. The bundles of the axons in the central nervous system forms tract. Okay, the central nervous system, we will discuss about the tracts. They are nothing but bundles of axons in the CNS. While the bundles of axons in the peripheral nervous system, they will form the fascicles. So, bundles of axons in the CNS is called tract and in the PNS they are called fascicles. Now, the axons originate from the axon hillock and they will conduct electrical impulses in the form of action potentials away from the cell body. How they will conduct these impulses? Through a process of sequential depolarization and repolarization. So about this, about the conduction of impulses, we will go in detail of this topic in electrical properties of nerve fibers. Okay. Now, these axon terminates in axon terminals. They are fine branches and they are called terminal arborizations. And each of these fine branch is capped with a terminal button. So, the terminal thing is enlarged. It is called terminal button. These specialized structures contain synaptic vesicles. These synaptic vesicles will store the neurotransmitter which is to be released into the synaptic cleft when the action potential reaches the axon terminal. So, what is this synaptic cleft? It is nothing but a small gap present between the two neurons. That is, two neurons. So, this is about axon. Coming to myelin sheath. The axons can be enveloped with a insulating layer of lipids and proteins that is called myelin sheath. This sheath will protect the axons and also prevents the loss of electrical charge during the transmission of action potentials, and thereby it will increase the speed of impulse transmission. So it will protect the axon and it will increase the speed of impulse transmission. Okay. So this myelin sheath is formed by specific types of glial cells. So we have glial cells also in the nervous system. They are supporting cells, and the myelin sheath is formed by one type of glial cells. So in the CNS, the oligodendrocytes will form myelin sheath. In the PNS, Schwann cells will form myelin sheath. Okay. So the myelination of PNS axons involves many Schwann cells. So many Schwann cells will form myelin sheath of the PNS axon. Okay. Here in the CNS, oligodendrocyte will form myelin sheath. But in what is the difference is one oligodendrocyte will form myelin sheath for many axons in the central nervous system. So these nerves are called myelinated nerve fibers. But we don't have myelin sheath for some axons. They are called unmyelinated nerve fibers. You can see here, this is the central nervous system neuron. This is the peripheral nervous system neuron. Here, this myelin sheath is formed by oligodendrocyte. Here, these in the PNS, many Schwann cells will form myelin sheath. So here you see, this is the peripheral nervous system neuron in which myelin sheath is formed. You see myelin sheath formation in the central nervous system and peripheral nervous system. This is the central nervous system neuron. Here oligodendrocyte will form myelin sheath for this neuron and this neuron also. Okay. Here in the PNS, how the myelin sheath is formed from the Schwann cell. First, the axon invaginates the Schwann cell. This is the axon. This is the Schwann cell. The axon invaginates the Schwann cell and further invaginates the Schwann cell later on. Then it forms a mesoaxon. Then this mesoaxon, okay, makes several turns around the axon. You see here, this mesoaxon, this is the mesoaxon. It takes several turns around the axon, okay, and forms the myelin sheath. Now coming to the neurolemma. This is the outermost cytoplasmic layer of Schwann cells which is overlying the myelin sheath is called neurolemma. It is important in the regeneration of the damaged peripheral axons. Okay. In contrast, in the CNS, the neurolemma is not present. So, there is limited regenerative capacity in the central nervous system neurons. So, neurolemma of the Schwann cells is important for the regeneration of the myelinated axons in the peripheral nervous system. Now coming to the nerve fibers classification. They are classified into different groups based on their myelination. That is, group A neurons are heavily myelinated, group B are moderately myelinated, and C are non-myelinated. Along myelinated axons, they are present nodes of Ranvier. What is this nodes of Ranvier? These are nothing but the gaps between the myelin sheath. What is the importance of these nodes? they will allow electrical impulses to jump from node to node. Okay. So, uh, nerve fibers classification in detail we will discuss at the last. Okay. So, here you should remember that group A neurons, B neurons and C neurons broadly. A are heavily myelinated, B are moderately myelinated and C are unmyelinated. In the myelinated axon, we have nodes of Ranvier. These are the gaps between the myelin sheath and it will allow electrical impulses to jump from one node to another node. So fast conduction of impulse can be possible. This type of conduction is called saltatory conduction because it appears that electrical impulses are jumping from one node to another node. And this is a small point. Myelin sheath gaps are more numerous in the peripheral nervous system than in the central nervous system. See here, it is myelin sheath. In between the myelin sheath, gaps are present. Those gaps are called node of Ranvier. So, here it is important for saltatory conduction in the myelinated nerve fibers. What is this? The depolarization only occurs in the gaps between the myelin sheaths. Okay. Why it is not occurring in this area? Because myelin layer prevents the movement of the sodium and potassium ions through the cell membrane, that is axonal cell membrane. So here depolarization is happening, then again here it is happening, then here it is happening. So it appears that the impulse is jumping from one node to another node. This is called saltatory conduction. It enables faster conduction in the nerve. And here the axons are not having endoplasmic reticulum, ribosomes, proteins and organelles needed for its growth. But these are synthesized in the cell body and they will transmit it to the axon via the axonal transport. Okay. And this axonal transport is facilitated by microtubules and intermediate filaments. These will form cytoskeletal tracks for the transportation of these substances. Okay. So microtubules and other intermediate filaments, they will form tracks. So that on the tracks, with the help of some components, these proteins and other required products are transported from cell body to the axon. Now coming to the overview, key facts about the parts of the neuron. Coming to first, cell body or soma, it is spherical or polygonal and it is present in the center of the neuron, central component of the neuron, and it is present in the center of the dendrites. The function is it integrates and it processes the signals and it also synthesizes the proteins and it is involved in the metabolic activities. The components present are nucleus, which is containing DNA, and cytoplasmic organelles such as smooth and rough endoplasmic reticulum, Golgi apparatus, microtubules, mitochondria, lysosomes are present and it ends in the axon hillock. Axon hillock is a specialized cone-shaped region of the cell body which forms the initial segment of the axon. It functions as the initiation of the impulses. That is, it is the site for the initiation of the action potentials. So, it is also called as trigger zone of the neuron. But here, there is a lack of large cytoplasmic organelles. But it contains high density of voltage gated sodium channels. Now coming to the dendrites, they are tree-like, short and tapering process of varying shape. What is the function? They will receive the synaptic signals. Okay. And they will translate it into electrical events. The components are similar to the cell body. That is, it is also having cell organelles. and it is also having neurotransmitter receptors. Okay, and it is having dendritic spines and shafts. Now coming to the axon, that is nerve fiber actually. It is a single long process arising from the axon hillock. It is for conduction of electrical impulses away from the cell body. This is covered by cell membrane called axolemma, and it is having cytoplasm called axoplasm. And in myelin fibers, it is covered with myelin sheath. And the gaps between the myelin sheath is called nodes of Ranvier. And terminally, this axon is terminated as terminal arborizations. And each arborization is a slender extension. It is having terminal boutons. That is somewhat expanded like things. And it is having my intermediate filaments and microtubules. But it is devoid of endoplasmic reticulum and ribosomes. Now coming to the zones of the neuron. Zones of the neuron. You have 4 zones. That is, you already know this. Receptor zone. So these are the functional zones. 4 functional zones are receptor zone. It is nothing but dendrites which receives the impulses. Then second is the site of origin of conducted impulse. Okay. So here the axon hillock. So this is the site of origin of conducted impulse. So this soma will process the information and decides whether the information has to be passed or not. Then axon hillock will generate the action potential. Then coming to the zone of all or none transmission, that is the axon. Okay. So here the signal transmission will happen in the all or none manner. Now coming to the zone of secretion of transmitter, that is axon terminals. Here the neurotransmitter will be released. So receptor zone, that is dendrites and cell body. Okay. So they will receive the signal and they will receive the signal and they will process the signal. Then signal will be integrated and there will be origin of the conducted impulse in the axon hillock. Then it is transmitted via the all or none transmission in the axon. Then there will be secretion of the neurotransmitter at the axon terminals. That is a zone of secretion of transmitter. That is receptor zone, site of origin of conducted impulse, zone of all or none transmission, and zone of secretion of transmitter. These are the 4 functional zones in the neuron. Now coming to the types of neurons. Types of neurons. So we can divide the neurons based on the number of poles or depending upon the functions and depending upon the length of the axon, we can divide the neurons. First coming to the types of neurons depending on the number of poles. It may be unipolar neuron, pseudo-unipolar neuron, bipolar or multipolar neurons. So this is the unipolar neuron. This is the pseudo-unipolar neuron. So here what is there? The unipolar neurons are present actually in the embryonic stage in the human beings. Okay. Now this is a pseudo-unipolar neuron, means it is starting as one. Okay, one. Then it is dividing into 2 branches. So that is pseudo-unipolar. It is in unipolar you have only one branch. It is in the pseudo-unipolar it is starting as one, but it is dividing into 2. So the pseudo-unipolar neurons, the example is primary sensory neurons. Primary sensory neurons, that is neurons which are conveying the impulses from sensory receptor to the spinal cord. Okay, from the sensory receptors to the spinal cord. Some sensory neurons are there which are carrying information. So those are pseudo-unipolar neurons. So this is the sensory receptor. For example, here skin is present. Okay. So these endings are present on the sensory receptor. They are carrying the information. And this is the cell body which is present in the dorsal root ganglion. And this is the central process. It is going to the spinal cord. Okay. Then coming to the bipolar neuron. So on either side of the soma, you have two extensions. For example, the neurons. These are mostly present in the special senses, that is in the vestibular ganglia, cochlear ganglia, and in the nasal olfactory epithelium, and in the retina. You have these bipolar cells. So where they are present? In the retina, okay, retinal layers, and in the nasal olfactory epithelium, and vestibular ganglia and cochlear ganglia. You have bipolar cells. Now coming to the multipolar cells. So the remaining all neurons are multipolar. So they are numerous. So here what is there from the soma? Many, that is axon and many dendrites are emerging. So this is multipolar neuron. So most vertebrate neurons, especially in the central nervous system, these are multipolar in type. So they have many poles. Now coming to the next type of neuron, it depending upon the function. It can be motor neuron or sensory neuron. So motor neuron is also called efferent cell, efferent neuron. So what it will do? It will carry motor impulse from the central nervous system to the peripheral effector organs. The effector organs can be muscles, glands or blood vessels. Okay. And these neurons have very long axons and short dendrites. Okay. So here this motor activity is coming from the upper side. So from the central nervous system to spinal cord, then spinal cord to the effector muscle. Then sensory neurons. These are afferent nerve cells. What they will do? They will carry sensory information from periphery to the central nervous system via the spinal cord. These neurons have short axons and long dendrites. Now coming to the other category of the neuron, it depending on the length of the axon. That is Golgi type 1, Golgi type 2 neurons. The Golgi type 1 neuron have long axon. Long axon. Okay. And Golgi type 2 neuron have short axon. So it depends on the length of the axons. So Golgi type 1 neuron, it is also known as projection neurons. Projection neurons. It is having long axon. So their cell body lies in the central nervous system and the axon can reach the remote peripheral organs. Okay. So in the motor neurons you have seen it has very long axons. Kada? Is it? Okay. They include the neuron forming peripheral nerves and tracts of brain and spinal cord. So as we have discussed, the motor neurons will come under Golgi type 1 neurons. Now coming to Golgi type 2, that is having short axons. And they are especially numerous in the cerebral cortex, cerebellar cortex and the retina. Okay. So Golgi type 2 neurons are mostly present in the cerebral and cerebellar cortex and the retina. Now coming to the neuroglia. So up to now we have discussed about the nerve. That is the functional zones in the nerve and what is the different structures present in the nerve and what is the function of each structure and what about the myelination in the nerves, okay, from which cells the myelin is formed and for what purpose it is formed, and we have seen the types of neurons based on the number of poles, function and length of the axons. Now coming to the neuroglia. These are the supporting cells. This term is coming from the Greek. That is neuroglia means glue. Glia means glue. So it is the glial cells, glue-like cells present in the nervous system. It is the diagram showing types of neuroglia. So in the central nervous system, we have four types of neuroglial cells: ependymal cells, oligodendrocytes, astrocytes, and microglia. In the peripheral nervous system, we have satellite cells and Schwann cells as a neuroglia. Now we will see each one by one. Neuroglia in the central nervous system, it can be classified as macroglia and microglia. Okay. So macroglia are further classified into oligodendrocytes and astrocytes. You see this oligodendrocytes function, you already know that it will form myelination in the central nervous system. Okay. So this oligodendrocyte will differentiate, that is, it is a oligodendrocyte progenitor cell. First it is a oligodendrocyte progenitor cell. Then it will form its a precursor, that is oligodendrocyte precursor. Then it will form pre-myelinating oligodendrocyte. Okay. Then it will formed into myelinating oligodendrocyte. Okay. From the progenitor cell to precursor, then pre-myelinating to myelinating. Then this myelinating oligodendrocyte can myelinate the axon. This is the oligodendrocyte. Okay. The oligodendrocyte will have rounded or pear-shaped body. It will have a round body or pear-shaped body. Okay. Then coming to the next macroglia, that is astrocytes. These can be fibrous astrocyte or protoplasmic astrocyte. Okay. So what is the difference between these two? That is, fibrous astrocytes are usually found in the white matter and they have long, thin, smooth processes with a few branches. And the cytoplasm is relatively thick because of the abundantly presence of the fibrils. Whereas protoplasmic astrocytes, they are usually found in the gray matter in a satellite position surrounding the neurons. And they have a thick, highly branched processes. And they will have thin watery cytoplasm. So these are the differences between the fibrous astrocytes, which are found in the white matter, and the protoplasmic astrocytes, which are usually found in the gray matter. Okay. So these astrocytes are actually star-shaped cells. They will give many processes. Okay. And these processes, when they will, these processes will sometimes come in relations with the blood vessels in relation to the surface of the brain. Okay. So this astrocyte, if the process is coming into contact with the blood vessels, it is called protoplasmic astrocyte. Now coming to microglia. So this is microglia. So here one important thing, the macroglia are large glial cells are ectodermal in origin. They are astrocytes and oligodendrocytes. The microglia, these are mesodermal in origin and they are actually formed from monocytes, okay, which are present in the brain. So these some monocytes settled in the brain, they will form microglia. Okay, these are the smallest neuroglial cells. Okay, and these will have flattened cell bodies and have short processes, and these are present more numerously in the gray matter. Okay, so what is the function? So function, it can present the antigen, that is acting as antigen presentation cells, and they can self-renew also. Some damaged microglia can go into apoptosis and some will renew. That is, self-renewal is possible, and they can produce cytokines and chemokines, and they can do phagocytosis. Okay, and they are important for immunosenescence, and it is important for synaptic pruning. Okay, and they will release trophic support for remyelination. So they will give support for remyelination. Okay, and they are also important for blood brain barrier permeability. So all these functions are done by microglia. Okay, whereas the macroglia, in that macroglia oligodendrocyte is forming myelin sheath, and astrocytes, they will perform some activities. What are these activities? They will regulate the synaptic activity, and they will regulate the metabolism of neurotransmitters, and they will also maintain blood brain barrier. Okay, and the important thing is they can take up potassium ions and GABA neurotransmitter, and helps in the maintenance of the concentration of these ions. Okay, so we have activities for both macroglia and microglia. These are the functions of the microglia, and I already told functions of the astrocytes and oligodendrocytes, which are included in the macroglia. Now coming to ependymal cells. These are the ependymal cells which are present in the ventricular system of the brain and spinal cord. So these are the cells which line the ventricular system of the brain and spinal cord. What is the function? It is involved in the production of cerebrospinal fluid, and it's a part of choroidal plexuses. Now coming to neuroglia in the peripheral nervous system. They are Schwann cells. Okay, Schwann cells. So these Schwann cells, you know that they will produce myelin sheath in the peripheral nervous system, and satellite cells. These satellite cells are present around the cell body of the neuron. Okay, in the peripheral nervous system. Now coming to overview of the neuroglial cell types and function. So in the central nervous system, we have astrocytes, oligodendrocytes, which are macroglia. So oligodendrocytes will form myelin. Astrocytes, they will maintain blood-brain barrier, and they will maintain level of the neurotransmitter at the synapse level, and they will regulate ion transport and metabolism and metabolic transport, and they will give metabolic support. Now coming to microglia, actually it provides the immune system in the brain and removes the dead cells and pathogens also by phagocytosis. Ependymal cells, they will line the ventricles and spinal cord, and they will produce CSF. Now coming to the PNS, the Schwann cells will produce myelin, and the satellite cells, they will surround the cell bodies and regulate neurotransmitter levels. Now coming to the peripheral nerve. You see this cross section of the peripheral nerve. So what is a nerve? It is a compact bundle of, it is formed by bundle of axons located outside the central nervous system. Is called a nerve. So what is a nerve actually? It is formed by compact arrangement of bundles of axons, and it is located outside the central nervous system. It is called nerve. In a nerve, the axons are arranged in different bundles called fasciculi. So you have already seen that bundles of axons in the peripheral nervous system, it is called fasciculi. Okay. So this is, you can see this is the axon. It is surrounded by myelin sheath. Okay. Each axon is surrounded by endoneurium. Endoneurium. And some axons will be surrounded by perineurium. So this is the one fascicle. Like this, we have many fascicles. These many fascicles are surrounded by epineurium. Okay, this is endoneurium, this is perineurium, and this is epineurium. So this is a cross section of the peripheral nerve. Okay, so each axon is surrounded. Each axon or nerve fiber. This is a nerve fiber is surrounded by endoneurium. Okay, so each then group of some axons is called fasciculi. Each fasciculus is surrounded by perineurium. Okay. Then this whole nerve is surrounded by epineurium. Now coming to the second topic in this video, that is biological activities. Okay, what are the biological activities of the nerve? First coming to the protein synthesis. Okay, in this biological activities we are going to discuss about the protein synthesis, exoplasmic transport, metabolism and heat production in the nerve fibers. So first coming to protein synthesis. This protein synthesis occurs in the soma and dendrites. Okay, with the help of nissel bodies. So these are the sites of protein synthesis. The nissel bodies are nothing but fragments of rough endoplasmic reticulum, and they are present in the soma or cell body, and some are also present in the base of the dendrites. You see, this is the nissel body, which is involved in the protein synthesis, and these are more present in the large active neurons such as spinal motor neurons. You can see more number of nissel bodies or nissel granules. Now, protein synthesis can also occur in all synaptic compartments also, including excitatory and inhibitory axon terminals. And the type of protein which is present the in the nerve terminal, it indicates the type of synapse and the strength of synapse. The transcription occurs in the nucleus, and it involves RNA polymerase. Okay, using this RNA polymerase, what it will do? It will attach to the DNA template and it will produce mRNA. Then this mRNA comes out of the nucleus and translation happens in the cytoplasm. With the help of ribosomes, tRNA, rRNA, what they will do? They will translate mRNA to the chain of amino acids. So, and forms the final protein molecule. So, this is the transcription and translation which are occurring in the process of formation of proteins. Mainly proteins are formed in the cell body and dendrites, and some can also be formed in the synaptic compartments, that is axon terminals. Now coming to the second biological activity, that is exoplasmic transport. Exoplasmic transport. The exoplasm is nothing but cytoplasm in the neuron. Okay, and it is in a constant motion. Okay, and this exoplasmic transport is essential for the nerve to perform its functions, because the axon cannot prepare its proteins etc., because it lacks rough endoplasmic reticulum, that is it lacks Nissus granules, and some materials should be transported to and fro. Okay, so this can be divided into rapid transport and slow transport, and this rapid transport can be anterograde or orthograde transport, that is from cell body to the axon terminals. Okay, cell body is considered, the part towards the cell body is considered as minus end, and part towards the axon terminal is considered as plus end. Okay, so this is performed by this kinesin molecule. Okay, this is kinesin molecule which is involved in anterograde transport. So it will attach the cargo here and the microtubule assembly will be formed first. Okay, for the transportation of the substances, the microtubules will assemble like this. This is the minus end, this is the plus end. So kinesin will move from minus end to plus end, that is towards the axon terminals. Okay, then coming to the retrograde transport, that is in this direction from axon terminals to the cell body. It is performed by dynein, cytoplasmic dynein. So it is attached with the cargo here and then they will go from plus to minus n. They will walk on the microtubules. So in the anterograde or orthograde transport, what are the materials transported? Okay, they can be mitochondria, small vesicles, actin and myosin can be transported. In the retrograde rapid transport, okay, some proteins and small molecules can be transported. You see, this is the second type of exoplasmic transport, that is slow transport. Always it occurs in the anterograde direction only, that is away from the cell body, and it is unidirectional. And here you can see the fast and slow transports. The fast component, that is fast anterograde transport. The average rate of this transport is 200 to 400 mm per day. What are the moving structures? Golgi derived vesicles and tubules. Okay. So in that vesicles and tubules, what is there? It is having synaptic vesicle proteins, kinesin, enzymes of neurotransmitter metabolism will be there. And fast retrograde transport, endosomes and lysosomes can be transported. And these are having some internalized membrane receptors, neurotrophins, and active lysosomal hydrolysis may be present in these structures. And coming to bidirectional transport, which is occurring 50 to 100 mm per day, mitochondria can be transported bidirectionally. And it is having cytochromes and enzymes of oxidative phosphorylation. Then coming to the slow transport. Slow component, which is occurring at the rate of 0.3 to 3 mm per day, neurofilaments and microtubules can be transported. In that, they may contain neurofilament proteins, tubulins, pectin and tau proteins. And coming to the slow component, which is occurring at the rate of 2 to 8 mm per day, here also they will transmit microtubules, supramolecular complexes of the cytosolic matrix. It may contain actin, clathrin, dynein, dynactin, glycolytic enzymes. Okay, so these are the axonal transport mechanisms. Now coming to next biological activity, that is metabolism and heat production in the nerve fiber. So like other cells, the metabolic activities also occurs in the nerve fibers, but they will occur at a very low level. So at rest what will happen? 70% of the total energy is required to maintain polarization of the membrane by the action of sodium potassium ATPase pump. So at rest also nerve needs energy. Why it needs energy? For maintenance of polarization of the membrane. This polarization of the membrane is done by sodium potassium pump. Okay. So this pump requires energy and this energy is derived from ATP. And this will release some heat, that is called resting heat. So 70% of the total energy is required to maintain polarization with the help of sodium potassium pump and it will release resting heat. Then what happens during activity? Okay. Pump means this is sodium potassium pump. So during activity what will happen? This sodium potassium pump activity will be increased and heat will be produced. Okay. So, for example, if sodium concentration is doubled, the pump activity must be increased by 8 folds. Okay. Then this will release some heat. The initial heat is released during action potential. Okay. So, during the transmission of action potential, initial heat is released. Then after the action potential is stopping, a recovery heat is released after the activity. It is 30 times the initial heat. The recovery heat will be 30 times greater than the initial heat. Okay. So there will be resting heat and initial heat, delayed or a recovery heat. Okay. It is 30 times the initial heat. And this stage is also lasts for 10 to 30 minutes. Okay. So this recovery stage is lasts for 30 minutes and it releases 30 times than the initial heat. This energy is supplied, this total energy is supplied by combustion of sugars and phospholipids. Okay, so ATP are derived from combustion of sugars and phospholipids. So during a nerve activity, the ATP and creatine phosphate will also be broken down. That is, they will undergo into hydrolysis and supply energy for the propagation of the nerve impulse. So this ATP and creatine phosphate are coming from sugars and phospholipids. And these are the heats which are produced in the metabolism of the nerve. That is resting heat, during activity, initial heat and recovery or delayed heat. Now coming to the physiological properties of nerve fibers. So now we are discussing about physiological properties. There are 7 physiological properties of the nerve fibers. What are they? Excitability, all or none response, refractory period, summation, accommodation, infatigability and conductivity. So these are the physiological properties of the nerve fibers. So because the nerve cell is an excitable tissue, okay, it is having low threshold for excitation. Therefore, the main properties of the nervous tissue are excitability and conductivity. So, the important ones are excitability and conductivity. Okay, and the remaining properties are secondary. So, first coming to excitability, that is the nerve fibers when stimulated, what will happen? They will produce waves of depolarization. That is called excitability. Then coming to conductivity, what will happen? The nerve fibers can conduct the electrical impulse along their entire length. That is conductivity. Then coming to all-or-none response. So, when stimulated, the nerve fiber either shows a response or it doesn't show a response. That is all-or-none phenomenon. Okay. So, there may be 0 response or 100% response. There is nothing between the 0 and 100%. That is all or none response. Coming to the refractory period. Okay. When nerve fibers is stimulated by 2 successive stimuli. Okay. 2 successive stimuli of more than threshold intensity. It responds to the first stimulus and it cannot respond to the second stimulus. Why? Because it is present in the refractory period. Okay. So, we are stimulating with 2 stimuli successively and immediately with the more threshold, but it is not showing any response for the second stimulus. Why? Because that nerve fiber is uh, present in the refractory period during when we are giving second stimulus. So that is called refractory period. Now coming to summation. Summation means if we are giving subthreshold stimuli repeatedly, these stimuli can be summated. So, if the interval between the two stimuli is less than 0.5 millisecond, then these can be summated. Although we are giving subthreshold stimuli, these can be summated and the nerve fiber will generate action potential. So, that is summation. Okay, there may be spatial summation or temporal summation. Temporal means time. It is we are giving the stimuli repeatedly in one place only, but repeatedly, successively. And the summation which is happening there is temporal summation. And coming to spatial summation, what is mean by spatial summation? We are stimulating the nerve at a uh, different places, but all these uh, different places at the same time, but these can be summated, these subthreshold stimuli can be summated, and the nerve fiber will give a response, that is spatial summation. Now coming to accommodation, that is the excitability of the nerve fiber is decreased when a constant current is passed through it. It is because of adaptation. Okay, that is nerve fibers will accommodate and their nerve endings will adapt when a constant current is passed. So, if a constant current is passing through the nerve fiber, it won't be excited. That is because of adaptation. So that phenomenon is called accommodation. Now coming to the infatigability. That is the nerve fibers cannot be fatigued even if it is stimulated continuously for a long time. That is called infatigability. These are the 7 physiological properties of the nerve fibers. Now coming to the last topic in this session, that is classification of the nerve fibers. We have 3 classifications. First one is letter classification of Erlanger and Gasser. Second one is numerical classification or sensory classification. Third one is susceptibility of nerve fibers to hypoxia, pressure and local anesthetics. It is also called physioclinical classification. So this can come as long question and these can also come as a short questions. So first coming to Erlanger-Glasser classification. The Joseph Erlanger, he was an American physiologist and he worked in the field of neuroscience. And he with Herbert Spencer Gasser, they identified several varieties of nerve fibers. And they have established a relationship between the conduction velocity based on the diameter of the nerve fiber. And they were awarded Nobel Prize in Physiology or Medicine in 1944 for these achievements. Here they have classified fiber types as these many: A alpha, A beta, A gamma, A delta, B fibers, C dorsal root fibers, C sympathetic fibers. Okay. So these A and B are myelinated, C are non-myelinated. Coming to the fiber diameter in micrometers, A alpha is having 12 to 20 microns and it has conduction velocity in meters per second as 70 to 120 meters per second. What is the main function of these fibers? They will carry proprioceptive impulses and somatic motor impulses. Okay. So they will have two functions. That is, afferent function is proprioception, bringing proprioception impulses from the periphery to the center. And the efferent function is motor function, that is motor nerve fibers or A alpha type of fibers. Now coming to the second type, that is A beta fibers. They are having 5 to 12 microns diameter and having velocity of 30 to 70 meters per second. Okay, and these are having only afferent activity, that is they will carry only touch and pressure sensations from the periphery to the center. Now coming to the A gamma, it is having 3 to 6 microns in diameter and conduction velocity of 15 to 30 meters per second. And these are having only efferent activity, that is motor activity to the muscle spindle. Motor activity to the muscle spindle. Now coming to the A delta, it is having 2 to 5 microns in diameter and conduction velocity of 12 to 30 meters per second. These A delta fibers, they are also having only afferent activity. That is, they will carry pain, cold and touch from the periphery to the center. Okay, so that is about A fiber types. A is further divided into alpha, beta, gamma and delta. Alpha is having both afferent and efferent activity. Beta and delta is having afferent activity, gamma is having more efferent activity. Now coming to the type B fibers, they are having less than 3 microns in diameter and velocity of 3 to 15 meters per second. Okay, they are also myelinated and it is also having only efferent activity, that is preganglionic autonomic fibers. So autonomic preganglionic fibers. Autonomic preganglionic fibers are type B fibers. So autonomic nervous system, you have sympathetic and parasympathetic. In sympathetic and parasympathetic, you have preganglionic fibers and postganglionic fibers. So all preganglionic fibers are type B fibers. Now coming to the type C fibers. These are non-myelinated fibers. Okay, these are non-myelinated fibers. So here, dorsal root fibers of C, they will carry somatic sensations, and C sympathetic fibers, they are postganglionic autonomic fibers, mainly sympathetic nervous system. That is postganglionic sympathetic fibers. Why it is specifying sympathetic fibers means, if you know that autonomic nervous system, the sympathetic fibers is having short segment of preganglionic fibers and a long, very long segment of postganglionic fibers. So it is type C fibers. And coming to the parasympathetic nervous system, it is having long preganglionic fibers and very short postganglionic fibers. So it is not mentioned specifically here. It will come detail in the discussion of the autonomic nervous system. Don't worry. Now coming to the numerical classification or sensory classification. So only sensory nerve fibers are classified, further classified in this classification. Number it is giving. So A alpha fiber type, they are further divided into 1A and 1B. So A alpha 1A is carrying sensory nerve fibers from the muscle spindle as annulospiral ending. And A alpha fiber type is forming 1B also. It is carrying information from the Golgi tendon organ. So it is coming, these topics will be discussed in detail in the discussion of the muscle spindle. Then A beta is called 2, type 2, number 2. It is carrying information from the flower spray ending of the muscle spindle and also touch and pressure sensations. Now coming to A delta, it is classified as number 3. It is carrying pain and cold receptors and some touch receptors also. Now coming to dorsal root C, it is classified under 4. That is, it will carry pain, temperature and other from other receptors. The information will be carrying through the dorsal root C or type number 4 nerve fibers. So here A alpha is further divided into 1A and 1B. A beta is called 2. A delta is called 3. Dorsal root C is called 4. So what is not there? A alpha is there, A beta is there, A gamma is not there. Okay, A gamma is only motor fibers, that is only efferent fibers. So A gamma is not there here. Now A delta is there. B is also not there. Dorsal root C is there. Okay. Now coming to the last classification, that is based on the relative susceptibility of the neurons. They are classified as A, B and C. So if the neuron is most susceptible for the hypoxia, it is classified as B. In intermediate susceptibility, and it is least susceptibility. And if a neuron is most susceptible to pressure, that is A. And if it is most susceptible to local anesthetics, it is classified as C. So in Erlangen-Gasser, you have fiber A, B, C. In that A, you have A alpha, A beta, A gamma, A delta. So that A is represented here. And B fibers also myelinated, they are represented here. And C, my non-myelinated, they are represented here. So in Erlanger-Gasser classification, A, B, C are there, no? So same here. These are the same. So most susceptible, intermediate susceptible, least susceptible to hypoxia, pressure and local anesthetics. Okay. So these are the 3 classifications of the nerve fibers. Okay. So in this video we have discussed about the nerve. That is, we are starting the nerve muscle physiology. In that we discussed about three broad headings in the nerve. That is functional anatomy of the nerve, biological activity, and physiological properties of the nerve fibers and the nerve fiber types. So these three classifications of the nerve fiber types are important. Nerve fiber types. Okay. So you should not get confused between the classification of the nerves and classification of the nerve fiber types. So in the classification of the nerves, we also classified that into three types. What are those three types? That is classification of the neurons or nerves. That is depending upon the poles, unipolar, bipolar, multipolar, depending upon the function motor, sensory, depending upon the length of the axon, Golgi type 1, Golgi type 2. That is classification of the neuron or a nerve. This is Erlanger gas classification, susceptibility classification, clinical classification. They are the classification of the nerve fiber types. Nerve fiber types. So, in one nerve, you may contain different types of nerve fibers. Then the next video is about electrical properties of the nerve fiber.